Dear students, let us see the module 2 replication in prokaryotes. These are the topics. This is the Michelson-Stahl experiment to prove that DNA replication is semi-conservative model. The enzymes and proteins involved in the replication are shown here. The reaction of replication is carried out by the DNA polymerase. There are different types of DNA polymerase in E. coli, polymerase 1, 2, and 3. And 1 and 3 are very important for replication to happen, and 2 is important for repair system. The comparative characteristics is given. The 1, 2, 3, all the three will do the proof editing function of DNA polymerase. It will go back and edit the mistake and then it move forward. This is a special function of DNA polymerase 1. So where once the replication is over, the fragments, Okazaki fragments are removed by this method, or if any nick is there, then that nick will be moved from that position to another position by replacing the existing DNA. It is done by DNA polymerase 1. Process of theta replication in E. coli starts with initiation, having ORI-C, initiation proteins, it forms the complex. Then initiation starts by forming a priming complex. Single strand binding protein stabilize, unwinding continues, and then the stress is relieved by topoisomerase 2. It starts by the synthesis of a primer sequence, and it proceeds in the direction of 5' to 3'. Elongation happens in the leading strand and lagging strand synthesis. Leading strand, once the primer is synthesized, it will proceed until the end, whereas in the lagging strand, it has to go just opposite to the direction of replication fork, but in the same direction of 5' to 3'. So at every regular interval, a primer is synthesized, then that will extend back. That is called lagging strand synthesis. It will lead to fragments called Okazaki fragments. And these two can be coordinated by a single enzyme by looping out the lagging strand template so that these two processes can go in parallel in one enzyme itself. Then once it is over, the fragments, Okazaki fragments are removed and nick will be sealed by ligase enzyme. The whole elongation process is given in brief. Then termination, when the replication fork meet each other at the opposite direction to the initiation or ORI-C, then there it forms the concatemer structure. The scatenated chromosome will be then separated and the stress will be relieved by DNA topoisomerase 4. Regulation, in the ORI-C itself, the sequence will be methylated at the N6 position of adenine at repeated GATC sequence. There are 14 of these sequences. If it is methylated, then it is ready for next replication. So it happens once the first replication is over, the site will be methylated, and this is an indication that it can go for the next replication. And it is until then it will be blocked. Other special types of replication. In viral DNA, we have sigma replication by covalent extension. Here a nick happens and that will be displaced, and the displaced template acts as the template for the leading strand synthesis, and then opposite to that a lagging strand synthesis occurs. And if we don't want double strand, the lagging strand synthesis is not required, then the protein will bind at the end of the nicked portion and it will loop back so that the lagging strand synthesis will be avoided. So examples are given for this looped sigma model also. And D-loop replication occurs in the eukaryotic DNA present in the mitochondria or chloroplast. And here the leading strand synthesis takes place and proceed, and lagging strand synthesis takes place little later. Until then, it gives a D shape, and the strand is displaced and it is giving a D shape. So it is called D-loop or displacement loop mode of replication. In virus example, if you look at the linear DNA, it circularize and form the ori region, and this ori region then will help for the synthesis by sigma mode of replication or theta mode of replication. And some virus, sometimes a protein will bind, and then that will facilitate the displacement of one strand, and thereby the other strand will be synthesized. And in one more strategy, this DNA will form a concatemer, and that concatemer will then break, and from there the replication can proceed to complete the end sequence synthesis. Special transcription happens, the RNA directed RNA synthesis, it is done by RNA replicase. The virus which is having RNA as genetic material, it can directly go for the translation process and protein synthesis. And for multiplication, then this will be then the single stranded RNA will be converted into another single stranded RNA with the help of RNA polymerase, RNA dependent RNA polymerase. And reverse transcriptase, certain virus, they have to integrate RNA virus, they have to integrate into the DNA. For that, the conversion of RNA to DNA happens. So this is done by a special enzyme called reverse transcriptase. So that will synthesize a complementary DNA sequence, and then the existing RNA will be degraded, and for that complementary sequence again one more sequence will be synthesized, double stranded DNA will be synthesized. This is called cDNA. This is what used in the genetic engineering concepts, and from RNA we can synthesize DNA. Thank you.